Thank you, Maurice. And I'd like to thank BenchSci for sponsoring this symposium today. And it's really impressive that all of you turned out in the middle of August to hear me and the other speakers. And I'd also like to say that it's really great to be back in Toronto. I haven't been here in several years, but I used to visit frequently. And now that I'm here, if my family wasn't back in the United States, I think I'd like to stay. Could someone sponsor me from the insanity going on in the United States? I swore I wasn't going to get political here, but that's it. Anyway, great to be here. So I am going to talk in the next 35 to 40 minutes about my view on reproducibility, and in particular, I mean, it's such an enormous topic that I'm really going to focus on my view of it specifically from where we can address the issue from a reagent standpoint. And I'm going to talk obviously about antibodies, but also about cell lines. And before I start, I also want to make the point that it's very easy, and I will do this, to criticize research right now, basic research, and the issues around reproducibility. But I want to emphasize, before I go down that rabbit hole, that there are solutions. And my organization, GBSI, is really focused on solutions, and I think BenchSci is as well. I think BenchSci is really creating a really important, really interesting tool that I think directly can begin to address the issues, the complex issues around reproducibility. So we do have a problem, and this is a slide we made a couple of years ago already, and it really takes a sort of snapshot of several studies that have been published over the last 5 years, 6 years, that in different ways, I should emphasize that, and in different, I think, approaches to the definition of what reproducibility is, which is also an interesting question that I don't have time to get into, but maybe we can in the Q&A. But really, I think perhaps was a tipping point, if you will, for this quote-unquote reproducibility crisis. And in particular, let's see if I get the pointer right here. In particular, it's really this study, this infamous Amgen study by Glenn Begley and Lee Ellis that was published in Nature. It was actually published as a comment, not as an actual research paper, that looked at 53 high-impact cancer biology papers and found that only 6 of the 53, so 11%, were reproducible based on the sort of general definition of what reproducibility is. And again, we could dig into that. There are other studies that showed similar results, and I think generally when we talk about reproducibility, and I'll show you a specific example in a minute, and I think we're being perhaps generous here, at least based on published work, we put it at about 50%. But this one, the Amgen paper was talking about 89% of the papers, or again, only 6 that were reproducible out of 53. And again, if there was a tipping point here, that could have been it. It got a tremendous amount of publicity, both in the scientific and lay press internationally, and I think that really sort of started a dialogue and a debate. Nature followed up just last year on a pretty detailed survey on reproducibility. There is, and I'm going to mention it at least at the beginning, there's a certain breathlessness to this crisis. Even the use of the word crisis has this sort of sensationalistic aspect to it, and Nature is guilty of that, just like the rest of us. 1,500 scientists lift the lid on reproducibility. Survey sheds light on the crisis rocking research. But, past the sensationalistic headlines, these results were pretty sobering. 52% of the respondents said that there was a very significant crisis in reproducibility, and 38% said, yeah, there's some stuff going on, but it's a slight crisis, whatever that means. But I think this result, this graph was, to me, much more, I think, revealing. And all of us, and I, as Maurice mentioned, I spent a lot of years in the laboratory, as a graduate student, as a postdoc, running a lab for 12 years at Sloan Kettering, and even in pharma. And we all know, and I was an editor for molecular and cellular biology for 10 years, and we all know that, in our own work or work that we review or work that we're familiar with from a competitor, that we've had issues reproducing experiments. And so, this shows you that, it's a pretty high response in terms of have you failed to reproduce an experiment. It doesn't say how many, but the number of people that have had this experience, and I'm sure you in the audience as well, I think tells a story. And I think one of the things, and again, I'll leave it to discussion, is, this is a problem that's been going on for years and years and years, and I guess the question is, why now are we sort of confronting this as a crisis, even though, we've all been familiar with it for years. But I think we have to own up to it, and if we don't, and when I say we, I mean the scientific community, because if we don't, it's going to be trouble, and there's already trouble. This was a cover. I think it was from 2015. I actually literally bought this magazine at a newsstand at 30th Street Station in Philadelphia, and that was the cover. And the article was obviously about the reproducibility crisis. This came, I believe, on the heels of the Amgen study. But when you make the cover of The Economist with the headline "How Science Goes Wrong," then I think that you got to worry. And there have been many other articles taking different angles on the reproducibility crisis. This is yet another example. This is a book that was just published this past spring by Richard Harris, who's the senior science correspondent for National Public Radio. I know Richard very well. He's been very supportive of GBSI, and he's a really careful reporter, but even he, although he will claim that it's his publisher, but even he felt victim to this sort of sensationalistic aspect to the reproducibility issue. I think rigor mortis is kind of a clever title, but the subtitle is really bad. How sloppy science creates worthless cures, crushes hopes, and wastes billions. Not good. So we've got to get a grip on this, and we've got to, as a community, begin to propose and execute on solutions. And I am not advocating, I'll just tell you this right off, I don't know what the structure is in Canada, but in the United States we have no regulatory authority that regulates basic research. It all kicks in once we get into the human clinical studies, and that's all done by the Food and Drug Administration. The NIH, which is the largest funder of research, I think still in the world, but that could change, sorry, is not a regulatory body. It's a funder. And we're not advocating by any stretch that rules and regulations kick in. We're advocating that we bring in best practices and standards, which I'll talk about, but at the level of the bench scientist. The challenge here is that the causes of irreproducibility are really complex. There is no quick fix to this by any stretch of the imagination. And when GBSI launched 5 years ago, we published a white paper, which you can download straight from our website as a PDF. And it's called The Case for Standards in Life Science Research, and I'll make the case for standards in a couple of minutes. But we also, we sort of deconstructed based on interviews of stakeholders from academia, industry, government, funding agencies, et cetera. We rank ordered, and you can quibble, and it's okay, about this rank order. We're going here from increasing to decreasing prevalence. But the point I really want to make with this slide is that there are multiple drivers for this issue around reproducibility, from how we report protocols to reagent variability and lack of validation, which I'm going to spend the rest of my time talking about, experimental bias, analytical technique differences. Statistics is a huge, huge issue for those kinds of experiments that involve complex statistical methodology. I think we can go on and on and on. I think the most interesting, or one of the interesting points from this slide, which makes me feel a little better, because sometimes when you look at this, it seems so complex that it's hopeless, is that actual scientific misconduct, which we're not talking about here, is actually quite rare in comparison to all these other drivers that contribute to irreproducibility. So although malfeasance is extremely serious, it's not something that when we approach reproducibility, we are really focused on in any way. And again, in comparators to the other drivers, it's actually quite rare, although very serious. The other aspect of reproducibility, before I get into the specifics, that really has been impactful, at least from my own personal experience, is this paper that we published now 2 years ago in PLOS Biology. It's the only paper I've been ever associated with that has economics in the title, or even in the paper. But it really created a stir, if you will. And I'm going to go through some of the data in a minute, but I'd also, somewhat defensively, because people get very, or people have gotten very aggressive about this paper, I want to make the point that since I'm not an economist by any stretch, I can't even deal with my own personal budget. We collaborated with two well-known economists from Boston University, Ian Cockburn and Tim Simcoe, who study, they really look at economic factors related to the use of standards in other industries, but we're very intrigued by the issues going on in biology, and in particular how it related to irreproducibility. And again, somewhat defensively, Tim Simcoe, when we were working on this paper, was actually on a leave from Boston University, and he was on the Council of Economic Advisors in the White House, and fortunately that was when President Obama was president. Anyway, so the way we approached this was sort of twofold. We did it sort of top-down and then bottom-up analysis. The top-down was remarkably simple, and I'm going to take you through that in just a couple of slides here. And this is all publicly available data. This was actually based on PhRMA, this lobbying agency in the United States that calculated the amount of money spent this is just the United States alone, on life science research in 2014. And it compiles industry, NIH, National Science Foundation, other foundations, academia, and it totals up to about $115 billion, and that was 3 years ago. And interestingly, it splits about 50-50 between preclinical and clinical research. We're going to just focus here on preclinical or basic research for the rest of this talk. So remember this $56 billion number, and then remember this slide that I showed you at the very beginning, where we are somewhat generously saying that although you add these up and you come up with, or you average these out and you come up with about a 64% irreproducibility rate, we actually put it at 50%, just because that was a nice even number and we were sort of being quite conservative here. So if you take that $56 billion that is being spent, at least in 2014, on preclinical research, and you apply a 50% irreproducibility rate to that, then you're talking about $28 billion that was being spent on on research that ultimately was not reproducible. There was a much more sophisticated calculation that the economists did that came remarkably close to this number, going from a bottom-up approach that I really don't have time to talk about, nor do I completely understand because I'm not an economist. But I will say that when we published this paper, I've published a lot of papers, a lot of scientific papers in my career. I've never experienced the kind of response, both positive and fairly hostile that we received because everybody just focused on that number. They didn't really read the rest of the paper. There are all kinds of interesting connections to other industries, proposals for how we approach the solution around reproducibility, but that focus on that $28 billion, as I said in a recent video, rustled some jimmies, if you will. But the other aspect of this paper, and this was a figure taken directly from it, is that we broke down, again, using sort of a meta-analysis, 4 aspects of research. It's kind of a higher view of that other figure I showed you where, there were like 10 or 15 drivers of irreproducibility. And again, based on some bottom-up meta-analyses, we tried to take that $28 billion number and assign proportionalities to the various drivers that we all experience in our own experiments in the lab. And the biggest, so protocols, of course, analysis and reporting, study design, and reagents and reference materials. And for us, it was really a focus on how we can begin to apply standards and best practices to biological reagents and reference materials where we could get really the biggest bang for our buck in addressing this issue. Because obviously, if you eliminated, just for the sake of this argument, $10 billion out of a $28 billion spend, that's a lot of money. So I'm going to talk about how we're approaching a greater awareness, a greater approach to how best practices around reagents can have an impact on reproducibility for the rest of this talk. And the first thing really to really approach here is that we all know, I certainly do, just from my days running a lab in academia, that there are no formal quality systems in place in academic research. And it's actually kind of interesting, having been in both academia and in industry, that the inverse is essentially true in pharma. Not that they don't have their own issues, which we could talk about another time, but it's almost an inverse in terms of formal quality systems and no formal quality systems. And our sort of, hypothesis for my organization is that standards, when applied the right way, can facilitate the alignment of best practices, reduce variance, and ultimately improve reproducibility. And here's sort of the conundrum. Standards have played a key role in almost every other industry and every other activity you can imagine and that you take for granted. In communications, in electronics, our cell phones, in our barcodes, which work everywhere in the world, an ATM card where you can go to, I don't know, Sierra Leone, and your, Wells Fargo, do we have Wells Fargo here? Banking card will work. Shipping containers, it's amazing. But what's amazing is that standards have completely been absent in basic biomedical research. And again, there may be good reasons for that, which I'm happy to talk about. But the question is, ultimately, do standards matter? And I think we've gotten to a point with the issues around reproducibility that the answer is yes. Otherwise, I wouldn't really be talking here. So, first of all, what are we talking about specifically when we talk about biological standards? Standards in general are an agreed upon way of doing something. They can be quantitative or they can actually be qualitative, where they describe a process. And likewise, there are material standards, which are highly characterized, actual physical substances, and I'll, again, give you an example of that in a minute, or written standards that really outline a consensus, a community consensus on how to do something along the line of practices, processes, and procedures. So before I go any further, here's the results of another poll that was published in The Scientist last year, and I think it sort of emphasizes the point I'm trying to make. So the question for this particular figure here was, in the past year, what products caused you issues with reproducibility. And the first three really, well, they're all reagents because they're product groups. But the first three are interesting. Animal models, primarily mouse models, which unfortunately I'm not going to talk about today. Antibodies, which I am going to talk about, and cell lines. So really, of the three biggies that in product groups that according to this survey are the biggest source of problems for reproducibility, I'm going to talk about the cell lines number 3 and antibodies number 2. And really, I know, we're here really to focus and celebrate BenchSci, which I share with you. I want to actually use the experience that we had around cell lines and then compare that to the challenges that we're all facing around antibodies. So just indulge me for the next couple of minutes about what I'm going to say about cell lines, because I think it'll be instructive in terms of what I talk about when we get to antibodies. So there are lots of challenges when you all use cell lines in the lab. There are issues around misidentified cell lines, which arise from cross-contamination, from just simple mislabeling, or even genetic instability, which is particularly prominent around cancer cell lines, which is what a lot of us have worked on in the laboratory. There are also issues around microbial contamination and culture heterogeneity. And this has really been, for the most part, ignored until the last couple of years. People, again, I can just, harken back to my own experiences running my own lab. If somebody needed a cell line and you could get it from, the lab down the hall, you do it. You would never think about necessarily issues around the identity or the authenticity of that cell line. Turns out that that's a big mistake, because misidentification and contamination are very, very common in the laboratory. And the last time a survey was done, it appeared that 15 to 36% of cell cultures that are in circulation have been misidentified. And there is a table that an organization of cell repositories has put together, an organization called ICLAC, where they have currently listing 475 misidentified cell lines. So you might want to check that out before you submit your next paper to Nature Cell Biology. And there's a huge cost to this, which I'll get to in a minute. And what is really disturbing is that the majority of researchers still don't authenticate their cell lines. This is a cartoon that one of our collaborators, Rich Neve, who at the time was a biologist, a cell biologist at Genentech. He's since gone to Gilead. He's a member of one of our task forces, and he turns out to be an amateur cartoonist. So my paper got accepted. Yeah, but the editor wants proof of identity for your cell line. Well, I did identify my cell line. The editor says writing the name on the vial doesn't count, and that's really sort of the level of where we're at even now. But it turns out that this is one of the rare examples where there actually is a standard. It's one of the few biological standards that exists. And the standard is using STR analysis or DNA fingerprinting for authenticating human cell lines. So I'm using this as an example because there are, as few as there are standards in biology, and they can have huge impacts. I'm not going to go through how you do STR analysis. It's PCR based. It's actually taken, ironically enough, from human forensic biology. One of the reasons it works is that the FBI in the United States created a huge database of STR profiles of all of us. And you can go back and use that for human cell lines to cross-reference the authenticity of a line. So as I mentioned, this technique was described as a formal standard. It was accepted by the international ISO ANSI International Standards Conferring Organization. It's called ASN 002. It was the second biological standard ever made in 2011, and it describes the authentication of human cell lines by STR. What's interesting is even today, and we've been pounding at this for now 4 or 5 years. There are guidelines in all the major journals, and at the very end of my talk, I'll refer to the National Institutes of Health guidelines on rigor and reproducibility. There's actually only one journal that requires evidence of authenticity of cell lines. So there are guidelines, but there are no requirements. And I think we still, despite the fact that there is this standard, it's easy. There are companies that will do it. You just send out, you just smear your cells on a piece of filter paper and stick it in the mail, and they'll do it for you for, you know, $120, $130, which is relatively cheap. There is a long way to go here, and that is borne out by a survey we actually did, GBSI actually did, that was published in Biotechniques in 2015. We actually had over 400 respondents. I'd never done a survey before. It is not easy to get 400 people to respond to a survey. We had to send it out to thousands of people. But the majority of them conducted basic research, they worked in academia, and they used culture cell lines. So these are people who really, you know, they're biologists, they're cell biologists. And what was shocking, and I'm just going to show you one result here, was that when we asked how frequently do you perform the following, you know, authentication, confirmation of species-related quality controls on your cell lines. And these are, I should mention that besides STR, which is the standard, you could do DNA sequencing, you can do fish analysis, you can do SNP, which has been described, you can even do isoenzymology and karyotyping, which are really, you know, old school. But in each case, and especially the STR, which I said is a standard, and there are companies that'll do it for you, 74% of the respondents said they never did it, which is really disappointing. So we have a lot of work still to do, and this was after we were already advocating pretty visibly around the issues around cell misidentification and how important it was to authenticate. And there's also a potential, we seem to come back to the dollar impact of all of this, because that's what people seem to respond to most viscerally. And again, we sort of did a back of the envelope calculation here. We took NIH Reporter and we searched for you know, grants that had been funded by NIH in 2015 that involved cell lines, and came up with the number of about, you know, close to $4 billion funded on cell-related basic research. If you take that 15 to 36% amount that had been done in a previous survey of cell lines that are misidentified, and apply it to this number, that's $1.3 billion. If standards reduced misidentified cells, didn't eliminate, you know, didn't bring it down to 0, but actually brought this number, 15 to 36, down to 5 to 10%, which I think is quite reasonable, you'd really gain a little bit over $1 billion from this $1.3 billion. So that is a lot of money, I think, just from authenticating cell lines. So there's a real economic gain here, and there's an approach. And yet we still have a long way to go in convincing the scientific community to do this. So let's talk about antibodies for the rest of the time I have here, because antibodies make cell authentication and the standard around cell authentication look really, really simple, which is kind of depressing, because I think that antibodies are much more impactful in terms of the number of people using them in the lab, the cost involved, and as I'll show you in a minute, the complexity of how to deal with the problems. So as the reproducibility crisis began to, you know, emerge and expand, there was a tremendous, there was a lot of focus, a lot of papers written, some quite visible, including these two in Nature, one sort of a comment by Manya Baker, who writes these features for Nature, and then another one by Andrew Bradbury, Andreas Pluckton, and a whole cast of others, signatories in the back around the need for standardizing antibodies. And as many of you know, if you work with antibodies, there really is, despite being among the most commonly used reagent in the laboratory, there are no universally accepted guidelines or standardized methods for validation. And there are, as we'll get into it, there are reasons for that. So we need standards to validate antibodies in an application-specific manner, and we'll talk about that in a minute. We need standards to ensure reproducibility of antibodies over time between manufacturers and even lots among manufacturers. And something I think is overlooked and something we're really engaged in is that training really is going to be a very, very key component here. Training of these best practices, training of these ways of validating, raising the awareness level, not just of students and postdocs, but even established investigators. And again, I'll have just a little bit to say about that at the very end. And I have this tendency, I already did it, earlier when I was talking about politics. When I talk to reporters, I kind of just go off and don't realize that they're quoting me, and it could be appear in a journal. But I've referred to this issue as the two-headed monster, and it's gotten quoted a bunch of times. And what I'm really referring to is that there are at least two aspects of it. There are, and we'll talk about it in a minute. One aspect is that there has been poor training in methodology that ensures that even the best antibodies from the best companies can be misused and not reproducible. But it's not all on you guys. The producers have a very, very clear issue in terms of their contribution to this mess because there's been very spotty QC, QA on antibodies. And another one of my quotes is that the industry is it's the Wild West, as we say in the United States. It is like you never really know what you're getting when you buy an antibody. And there are a few, and we know who they are, really companies who really care about quality, but the rest, good luck to you. So we did two things to begin to approach this issue. We kind of took a page from our experience with cell authentication, and we did a survey. And the results of the survey are much more nuanced and complex than the cell authentication survey. But actually, we did this survey at the same time we did the cell authentication survey, and we gave people the option to do one or both. So you could choose cells, antibodies, or do both. And here we got 504 respondents. Again, the same breakdown of basic research in academia, and we published it in a later issue of Biotechniques. And there are really only two things, because it is a little bit more nuanced, but it was also sobering. And that is, in response to the question, what are the barriers to performing antibody validation? The thing that really struck us is that, and we broke it down, the answer down to years of experience in the laboratory, was that 27, sorry guys, sorry of the young people in the audience, but 27% of people with 5 years or less experience in the lab said that they don't see the necessity of validating antibodies. And likewise, similarly, in terms of years of experience, I don't have my research antibodies validated. Again, the youngest cohort answered, a third of them said, I just don't see the necessity. So that to us shows that there are issues around an awareness level, an awareness around training. You don't just get the antibody from the company, take it out of the tube and, do your Western blot or immunoprecipitation. And just to belabor the point, and these are slides that were provided to us by Cell Signaling Technologies, CST, and of course they're making the point, and why wouldn't they, that user proficiency, you as the user is very important, because depending on how you fix these cells and then do cytochemistry to this antibody to BCL-XL, you can get very, very different results based on the fixative. And the same as, I don't know why I show this slide, I think it's just really pretty. And, but again, formaldehyde fix versus a methanol fix gives you a really, really different result. So, CST and Thermo and, they're gonna blame you guys for not handling their precious beautiful antibody correctly. But they're not getting off the hook here. The producers have tremendous spotty QC, QA antibodies, and they know it, and some of them care about it, and some of them don't. And there are multiple issues, some of which I was completely unaware of until I got into this. We know that quality varies from company to company, but what I didn't realize is that, and we know that there are unclear, unknown, and redundant sources. Many don't validate, many don't provide validation systems, many have poor QC. But what I didn't realize until we really dug into this at a meeting at Asilomar last year is that the market is plagued by resellers. So there are actually a vanishing few number of actual producer companies, but there are a lot of companies that resell those antibodies. Why is that a problem? Because they often rename the antibodies, and they often give a completely different lot number to those antibodies, so there's no way for you as the user to trace the origins of this antibody, which I think is completely crazy and ridiculous, and something we're trying to address, and I think BenchSci, with their super cool technology, could actually have some major impacts on. So we decided we had to get everybody in the room together and really begin to deal with this issue. So we had a meeting at Asilomar, and Asilomar has its own sort of vibe because that was where the first recombinant DNA meeting with the giants of molecular biology occurred in the 70s. So we actually very intentionally picked Asilomar, which is not necessarily easy to get to. It's south of Monterey, but it's beautiful. And we had several goals for this workshop. We wanted to begin to create validation standards, which other people have tried, and they're pretty much ignored by the research community. And I'll tell you how we think our approach is very, very different. We wanted to define the responsibilities of the producers and engage the suppliers. So this whole QA/QC system really has to get under control. And then we talked a lot about, actually the entire second day, it was a 2-day meeting, we talked a lot about implementation, because it's one thing to come up with guidelines and standards, but if they're not implemented correctly, they're just never going to get any kind of traction. So we had journal editors there, we had funders there, we had producer company reps there, and we had academic thought leaders there. And we also, by the way, talked about new technologies, including recombinant antibodies, which are heavily, I think have real promise, but have a long way to go. So I already mentioned our breakdown of who attended this meeting. It was over 2 days, and it was really great. And we came away with three goals, post-workshop goals, all of which are pretty ambitious. One is to create guidelines and scoring, and I'll tell you about that in some detail in a minute. The other is to create an industry consortium of companies where GBSI would sort of be the facilitator of this, but it would allow competitor companies to sit down and talk about their issues and their initiatives in ways that they never did before. And then I mentioned training already, and we think that training is going to be an equally key component to this. So we're working on all 3 of these now. But what I want to talk about, and what I think is really the most exciting and also the most work, is that when we entered Asilomar, we talked about guidelines. And I already mentioned to you that there have been several initiatives and published guidelines that are out there. You can just, go Google it after this meeting. They're there, but they're ignored. And so one of the initiatives that was advocated by a couple of very convincing people at Asilomar was to bring a scoring component to this. And that's what we've been working on since Asilomar. It's now August. We've been working on this since last November. And the way we had to approach this is, and this is one of the challenges I think about working on antibodies, is that it's not a one-size-fits-all guidelines or scoring. Every application that you use is going to have its own unique characteristics and properties. So as everybody knows, an antibody that is really great for a Western blot may be really bad for IP or for flow. And so each one of these applications, and we identified the 7 most common, has to have its own set of guidelines and its own set of scoring. So we formed 7 working groups of 6 to 7 people, and they've been working on these guidelines in an application-specific fashion. But it's really the scoring that we think is going to be the difference maker here. And what we're and it's really early, and I'm going to show you on the next slide a screenshot of what we have in mind. But the idea is to assign specific points to specific characteristics in 3 general categories that ultimately add up to 100 points. So the absolute perfect antibody, which I'm not sure exists, would score on-target, off-target, and technical properties that would equal 100 points. And I just want to tell you that there are people who are pushing back on this, but we're moving forward. There are companies, quite frankly, who are reserved and apprehensive about this, and I do think they have some legitimate concerns, which I'm happy to talk about in the discussion. But the idea would be to come up with a score sheet that would, again, identify various aspects of on-target and off-target properties. This would be within the component of an endogenously expressed protein, which would get the maximum score. An overexpressed protein that the antibody would pick up would obviously be a little easier and wouldn't get the maximum score. And this would be all online, and essentially you could do something like this if you had all the data, big point, in 15 minutes or so. So I'm wrapping it up. So this is our timeline. We're going to try to alpha test this in the next month or so because obviously we want to get this right. We'll come back and refine the scorecards based on feedback from alpha testing. Then we actually want to do a beta test with the public. We want to bring this out on a limited basis for a month and have some feedback, some public feedback. Then come back again and refine, and then ultimately publish this, hopefully in a high-profile journal, and then host it on some really cool website where both companies and users can do their scoring, and then those scores would be posted. So this is a lot more. I know that there are sort of many websites who are putting, sort of reviewer comments a la Amazon. Those exist. This is actually going to score and validate an antibody at the same time and try to put some quantitation in some granularity to the properties of an antibody. I want to just finish up. I told you, because I don't want to go any longer, I told you about this industry consortium. We're starting, here's the list of our leader companies, and we can talk about that later. The last thing, in the last literally 2 minutes, is that we really need to, there are some very practical solutions to addressing the issue of reproducibility, especially in the reagent space, okay? So I've talked about validation, and I think that there needs to be a broad adoption by vendors to sell only validated reagents, and perhaps a whole certification system has to come into play, which GBSI is interested in exploring. We need implementation through funders and journals, because that's the way we work. There is no regulatory body here. So, in academia, it's really ultimately the funders and journals that sort of carry the carrot and the stick here. And we really need to incentivize new tools and approaches. And then finally, in the last minute, I've been alluding to training programs. We've actually been directly engaged in 2 training, very specific training programs, both of which have been funded by the National Institutes of Health. One is a video, an interactive training set of training modules that teaches a user how to authenticate cell lines and to practice good cell practices, and it's not one of those boring, training videos that you can't wait to get through. And we're actually in the last 6 months of 3 months of putting this together, and we partnered with UCSF, the American Society for Cell Biology, and the University of New Mexico on this. And then the last training initiative is around experimental design. And we just got this grant. It's a $2.4 million grant, which we're really excited about. And it's really to, I think we should have gotten the grant just by coming up with this acronym, PREPARED, Producing Reproducible Experiments by Promoting Reverse Experimental Design. So we're starting with the result. And in this case, we're looking at imaging and high-throughput screening and disease modeling through our partners at Harvard and Vanderbilt. And we're working backwards to teach how to design an experiment. So that's just launching now, and we're really very, very excited about that. And then the last slide is that things are changing. And in the United States, NIH, which I have found, I mean, I was a recipient of many NIH grants in my career, but now I'm kind of on the sort of policy side, and I see how slowly things move. But NIH is hearing this, and they're trying in their own way to respond. And in January of 2016, they initiated this whole initiative around quote-unquote rigor and reproducibility. And part of it, it's a long, involved set of criteria, but there is a very specific area around where the applicant and also the reviewer has to analyze how key biological and also chemical reagents are authenticated. How that is assessed in a grant review is a whole nother question, but at least it's now out there, and NIH is beginning to move in the right direction, albeit slowly. So in the very last slide, I just want to sort of make a pitch for my little organization, GBSI. We by definition have to partner with you guys, with the scientific community, whether it's academic institutions, industry, professional organization, funders, journals. We're not doing this in a vacuum. It's all by virtue of advocating and working with you all. So it's been great to be here, and I don't know if there's time for questions now, but thank you.
So I'm sure you'll enjoy the presentation, and we'll have microphones on the side. If you guys have any questions, we have about 10 minutes, and we will have a 5-minute break until 11 o'clock. Then we'll start our second talk. So if you have any question, please feel free to raise your hand. Any question at all?
Yeah.
What are some reasons why the book Rigor Mortis is hyperbolic? I guess exaggerating the...
Oh, well, my comment was actually, so the question was, why is the book Rigor Mortis exaggerating? I actually was just referring to the title. I was asked to write a book review for Science Magazine on Rigor Mortis. It's the first book review I've ever written, which was actually a lot of fun. And it's really actually a very, very carefully written and researched book, which is the point I made in the book review. It was the title that I was really kind of getting on top of. So the book is actually very good and very careful, and actually points to something I didn't talk about, which is, but I alluded to, which is, part of the reason we're kind of in the mess that we're in, at least in the United States, is this issue, and this was a phrase that I first heard from Glenn Begley, the guy who did the Amgen study, of perverse incentives that are really driving research today. And I think rigor mortis really makes a very powerful point of that over and over again, which is that, you all, me as an academic researcher, our entire careers and jobs, careers, money really is based on our success in getting funding and publishing papers, which of course are also interrelated. And a lot of what drives, I think, a lot of the issues that are, really coming up as drivers for irreproducibility ultimately really stem from these perverse incentives that are in play in terms of how you succeed. in academic research. And nobody can argue that in my mind. And I thought the book made a really good point of that, and that was something I really focused on in my book review in Science. Yes.
So while it's pretty intuitive that following guidelines or standards would improve reproducibility, has there actually been research done to confirm this, and whether which parts of specific parts of standards?
Yeah, we'd love to do that. I think we're planning in the next year or two to somehow, I guess you'd probably do it through surveys, but we'd like to look at that. So the hope is that, there definitely has been an increased awareness. Just look at, people in this room here. But I think that is tempered increased awareness, and even, initiatives by NIH and also journals. Journals are now asking, my friends who are still in the labs are complaining to me that, the burden of everything else they have to do has now been increased by the checklists that the journals are asking for now. All those things, I think, will gradually, at least, begin slowly, gradually to increase the awareness level. But I think you're right. I think it would be great to somehow quantitate that. Too soon.
So
yes?
If data in a paper is found to be not reproducible after it's published, is there any mechanism to bring that information out to people who may be doing research based on those results?
Yes.
That would totally invalidate anything they're doing?
Yeah, that's a great question. So there, for years and years and years, there's been this process of, that journal, the onus is on the journal, and the onus is on the journal editors to retract or ask the authors to retract. So far, what we've seen and what I read and what journals have told me is that the only retractions that really typically take place are around, scientific misconduct, where there's really evidence of data manipulation. And that's where the process, a very, slow, probably for good reasons, process to retract papers. The problem that you're alluding to is that almost none of that occurs around, issues of reproducibility that are based on most of the stuff we've talked about. So all those papers are still, one of the most egregious examples of that is that despite the fact that there are cell lines that have been known for years to be misidentified, there are clear examples of those cell lines still being used and published in journals. So I think I think that unfortunately the literature is littered with irreproducible data that I'm not sure. I think we just have to sort of go forward now rather than clean up the mess behind us, which is kind of depressing. But yes.
Hey, so I'm a PhD student, one of the younger generations, but so there are companies who are notorious for bad quality antibodies.
Oh yeah.
And what I kind of find interesting is that you stated also that every single company advertises quality, and the same goes for these notorious companies. And what I'm curious about is that you see in commercialization or in any type of advertising, there's usually a sort of policy or a governing body that induces penalty upon misleading advertisements. And I'm wondering how much is that being applied in terms of research products?
That's a really interesting question. I would say zero, but it's an interesting idea because, as I said, there's no regulatory body in terms of, overseeing quality, but sort of misinformation or around advertising is an interesting angle that hasn't really been discussed, at least in the circles I've spoken to. And that's a very, very interesting approach that somebody should think about. But I also think, and I'll give a plug for our scores, is that once people start scoring and those scores get out there, I think, some of these companies, and we know who they are, you know who they are, they're not going to be able to, unless they cheat. And one of the concerns, quite frankly, and we were talking about this earlier, one of the concerns of the companies, especially the quality companies, is that the less scrupulous companies will game the system. They'll game the scoring. In other words, they'll cheat, and so they're very concerned about that, and we have to really think carefully about how to approach that. But if we can, once those scores are out there, they're not going to be able to escape the reality. But that's a very interesting point.
Hi, in follow-up to the gentleman's question about retraction, not necessarily everything that is irreproducible is wrong.
Yes.
Sometimes the materials and methods are just not extensive enough to be able to reproduce it. So how do you, GBSI or somebody else, start putting pressure on the journals to insist on extensive methods and materials?
I'm so glad that's a really, really good point. And actually, one of the points we made in that economics paper that was completely overlooked is that even papers that are not reproducible actually often have important information and reproducible components to it. So you can't throw the entire baby out with the bathwater. But to your point specifically, journals actually are beginning. That is one tangible area where journals are beginning to respond. Even the one-name journals where the methods. We used to save on words because we'd stick it. We'd create these super short methods sections. They're now allowing unlimited descriptions of methods, and I think that is very, very important. And I think that in some of these limited reproducibility initiatives where people have literally tried to reproduce papers, one of the biggest hurdles has been a dearth of description of the methods and the reagents. So there is tangible progress there.
Yeah, but there has to be pressure then on the authors to actually give a complete protocol, because the buffer components can make a huge difference.
Right, and I think journals are doing that now. I think they're asking their reviewers to specifically look at that. I think what concerns me, having been an editor and a reviewer for more years than I really want to remember, is that a lot of this is falling back on the reviewers who are, just trying to, that's all voluntary primarily. But there is some tangible movement there. Yes.
Hi. My question relates to more nascent areas of research, such as microbiome. Wondering if you've done any work in areas like that where it's not immediately clear what standards should be employed, because we're still trying to figure out the fields. Do you have any recommended approaches for those fields? Have you done any work in that area?
So we haven't done any specific work in those areas, but I think the microbiome, even next generation sequencing. I mean, just about stem cell biology. These are all areas that we're actually very interested in. And I think a lot of these general concepts can be applied to, but we haven't done anything tangible. I'll just give a plug in the last minute. We're actually organizing a one-day meeting in San Francisco on lab automation. I actually think that although previously automated tools, TCAN robots have been out of the affordability range of any academic investigator, there are some really cool companies coming up with very affordable, even miniaturized automated pipetters, if you will, which I think could have also in a sort of simple, intuitive way an impact on reproducibility. So we're sponsoring a meeting in October in San Francisco on that.